Good afternoon, everybody. Thanks for coming. This is a different venue for us for the Dean's Lecture because there's so many activities on at the school, but it's a great venue to have today's talk. As you probably all know, being appointed or promoted to the rank of professor at Johns Hopkins is not an easy task. And it's something that although I'm jaded about many things, I'm not jaded about this. I mean, it's really a great accomplishment, and that's why we have these Dean's Lectures to recognize people who have been appointed or promoted to that rank. And it's a decision, I want to make clear, that's not made by me or the faculty or the chairs or the A&P committee. It's really a decision made by peers around the world who we solicit input and a review, and they write. And that's why David Sullivan was, I guess, promoted last year to the rank of professor because of the outstanding reviews he got from people in the field of malaria. Malaria is not an easy field. It's divisive. There's people working in different areas, but the evaluations of David's were really outstanding. So let me tell you a little bit about him. You probably know all this, but I'll start. You know, he went undergrad to the University of Virginia and then went to the University of Alabama at Birmingham for medical school, and then left there to go to Washington University in St. Louis to do a residency in medicine and then a fellowship in infectious disease, and then was recruited here by Diane Griffin before we had the Malaria Research Institute to work in malaria. So he's probably now one of the longest surviving members of MMI because that department's expanded so much since then. Yeah, huh? Clive Schiff. Clive Schiff is there, and Clive's over there. So you and Clive. And because I get to interact with the Malaria Research Institute Advisory Board every year, I know probably more about the research in that group than I do about many of the other groups in the school. And on that board is Dan Goldberg, who was David's mentor at WashU. And so I just want to, and again, you're going to talk, I see, about iron and about drug development, but David has two passions. And for those of you who haven't read his faculty page, it's really a great faculty page. And he talks about when he treated a patient when he was a resident at WashU with malaria, a patient from Nigeria, and that's what really sparked his interest. And that interest is coupled with his interest in pharmacology and drugs and on drug development, and that's what he's going to talk to us about today. So I think malaria is an area that we know just has a great burden of disease around the world. We don't know exactly how many people die, but it's close to 1 million. Many of those people who die are infants who get malaria for the first time and get cerebral malaria and die. And sometimes when we're here in this environment, I think malaria becomes an abstract concept, especially for people who practice medicine here, because it's basically malaria is a board exam question. That's what it is. But many of you have worked in malaria, many of you have been abroad, and you know that it's not an abstract concept. It's really one of the plagues of humanity. So we're really grateful for the Malaria Research Institute, but especially for the work that David brings in trying to develop new therapies. So please join me in congratulating and welcoming David Sullivan.
Thank you, Dean Clagg. Thanks. I see many friends and colleagues. My wife and 2 sons, Ben and Ronan, also here. Thanks for all coming. So today is also World AIDS Day. So where does malaria fit in to all the other public health preventable diseases? Actually, back to Dean Clagg's area, hypertension claims the most deaths amongst public health preventable diseases, with around 9 million, and we're still only controlling about 10% of the people who have hypertension. Cancer is second on the list with about 8 million deaths, although smoking gets sort of a double count. There's about 1.5 million people who die from cancer each year worldwide, which would go under the cancer deaths, but also attributed in the smoking deaths. So the 3 top infectious diseases on this World AIDS Day includes malaria and TB, about 3 million deaths each year. We also know that if we were able to expand our vaccination programs, we could probably avert another 1.5 million deaths. And interesting enough, especially since I've seen it in India and Bangladesh and in other places, motor vehicle crashes still kill more people than any of the single infectious disease killers. And the latest tally from the malaria deaths, although Dean Clagg is right, we still can't count the number of people that die and have malaria each year, but it's estimated that we're at approximately 600,000 cases of death and 200 million cases each year. So what is this malaria? Strict definition, Hippocrates, it's fevers. Sorry, the pointer is not working as well here. You can see a large spleen, someone that is anemic. It's a typical cyclic fever pattern. But so all the malaria, the 5 malaria species affecting humans have that pattern, but P. falciparum causes more lethal disease. Here's a picture or cartoon of children in Africa which bear the brunt of the deaths fearing the mosquito. Interesting enough, in the news, Pope Francis was visiting Kenya. He was lauded for riding in an open vehicle, but was asked, aren't you afraid of things? And he said, no, what I'm most afraid of now is the mosquito. And so he's pretty smart. He's right. The mosquito is a killer. So where are we on preventing malaria deaths now? This lead author, Christopher Murray, had published 40 years before this. He was on the famous Murray paper in 1978. And although he was last author on that one, he sort of published, he's now an epidemiologist in Seattle and has done a lot of good scholarly studies on following deaths. And so here in this graph, you can sort of see from about 1990 is where we sort of had widespread chloroquine resistance in Africa, with chloroquine still being used. And that sort of is the uptrend in total malaria deaths. So about 2005, there's been a decrease in malaria deaths, although malaria cases have stayed about the same. And so and again, most of the deaths in red are children under age 5. As Phil Tuma, our colleague, says, malaria is the dream stealer because it robs parents of the dreams that they have for their kids because it kills. And I guess that's part of why I work on the disease, and part of why we have the Malaria Research Institute is because we know it's a disease which kills kids. So the main ways for malaria control are vaccines. To quote George Bernard Shaw, stimulate the phagocytes, drugs are a delusion. Ronald Ross, who got the Nobel Prize for implicating the mosquito, had worked for about 20 years in India, and he said it was scrupulous use of the bed net, how he avoided malaria. And then Paul Ehrlich, about the same time in the early 1900s, sort of began with chemotherapy. And so today, we're spending worldwide about $2.6 billion for malaria control, mainly for insecticide-treated nets, the Ronald Ross way, indoor residual spraying, and also money on diagnosis and treatment by itself. Everyone asked me about the malaria vaccine, so I'm just going to digress a minute to sort of give you an update on where we are. And it's about like Waiting for Godot. Actually, if you read the book, the characters had a rough time waiting for Godot. They didn't fare very well. I think we're doing a little bit better now. But as far as the malaria vaccine, how does it stack up to other vaccines? Chickenpox, a single dose, 85% protection from any form of chickenpox. 2 doses, 98% effective. Mumps, 2 doses are 90% effective. influenza vaccines, a single dose given once a year with matching vaccine to circulating influenza viruses. It has about a 60% efficacy for individuals younger than age 65 when the vaccine corresponds to circulating strains. We have a TB vaccine, but it doesn't really prevent infection reactivation. It really just prevents severe disease, TB meningitis or disseminated TB, but we're still using it. So GSK and Gates and others we have made progress on the RTSS, repeat region of a circumsporozoite protein, has a T cell epitope and 2 hepatitis B S antigens to make it immunogenic. When it was first tried in a small human challenge study, 6 out of 7 were protected. Although when it went to Africa in 2009 with 3 doses, it only showed a 50% efficacy at that time, and even less in children that were less than 3 months old. And a loose definition of malaria. So it didn't totally protect from any parasitemia, but just from high parasitemia and clinical disease. Just 2 weeks ago, in New England Journal, there was one article that sort of unwound some of the lower efficacy, similar to influenza, that the big take-home was if you have most 90% of the isolates throughout Africa, in this study, it was 8,000 kids, 2,000 malaria cases, but 90%, almost 1,800, 1,900 cases, were a mismatch to the vaccine strain. And in those mismatched cases, the efficacy was only 30% with 3 doses. So that's where we are. There's still debate, controversy over should we roll it out with this efficacy or not. GSK and Gates still want to prevent a single malaria case, and that would be worth the effort. And I agree. The best parasite is a dead parasite, and this will contribute to that. But it certainly did not account for the vaccine for a decreased trend in parasites. What about bed nets? In sub-Saharan Africa, where most of malaria is, bed nets really didn't start getting rolled out even until about 2010. So we already had a decrease in malaria cases before bed nets were really widespread. I'd like to attribute it to diagnosis and drugs, but although time-wise, it's more closely with this 2005 trend in malaria deaths, with an increase in gray as in sub-Saharan Africa, the number of diagnostic tests, and then a big increase in replacement of drug-resistant chloroquine therapy with effective artemisinin therapy. But truth be told, it's probably some environmental causes that are also accounting some dry spells, a lack of mosquitoes, in addition to nets and drugs and diagnostics all at once that account for that decreased trend in malaria by itself. Our 2 main drugs are quinolines, which come from a bark, and artemisinins, which also come from a plant. And of our drug classes, 3 out of the 5 main ones all target iron or heme, and that's sort of how I became more interested in how drugs work and about mechanisms of resistance by itself. But it also started a long time ago. I grew up about a mile from this statue in Birmingham, Alabama. It was on a steep hillside. If I was able to ride my bike up the hillside, the guard would let me in for free, and I could go take a look at Birmingham. So I think Birmingham did not exist before the Civil War, had all the makings for steel, and so I guess iron was sort of on the back of my mind growing up, tabled for many years. My two sons, Ronan and Ben, can tell me about the chemistry of iron, that it's able to conduct electricity and heat. It's 10th most abundant element in the whole universe, although the Earth has, it's the most abundant in Earth with 90% of iron being in the core. On the crust where we live, it's only about 5% by weight, 2% by total moles. Most of it is not accessible in hematite ores, but free iron, ferrous iron, is a necessary trace element used by all living organisms, except for, as Val Collada has demonstrated, that Borriella uses manganese rather than iron, but most all other organisms require iron. It's a double-edged sword in biology. It's beneficial, used for enzymes, hemoglobin and oxygen binding, required for energy production, required for DNA synthesis. But at the same time, it's toxic. It can generate free radicals, can inhibit enzymes as when it's in a free state, and it's actually used by white blood cells to kill bacteria and cancer cells. The chemistry of that killing is Fenton chemistry or Haber-Weiss chemistry that generates the O2 hydroxyl radical that ends up damaging proteins and peroxidizes lipids. And so this is sort of the toxic side of iron by itself. So what in the world is the malaria parasite doing inhabiting a red blood cell with 20 millimolar heme iron and lots of oxygen? So it just goes right to the thick of the battle with cleaves hemoglobin. Oxygen is released, iron, heme is released, and so that could just generate, if you were a white blood cell, this would be just what you want to do, but it's got to sort of survive in that environment. So it does take it, here up on the, on your upper right is transmission EM of the malaria parasite, which has gobbled up about 80% of the hemoglobin. There's the lysosome-like with the dark black area being the hemozoin, and which makes a, so the parasite has ended up making a crystal inside of itself. So crystallographers have it easy. They have pure proteins, and they slowly come up with 1,000 different ways to get rid of water so they can make a crystal out of something. But the malaria parasite has to make crystals inside of itself with lots of proteins, lots of other things around. And that was part of the problem or the puzzle that I was working on 20 years ago. Parasite also lacks heme oxygenase, which our cells use to degrade heme, and although it's out of the pot and into the fire because you got to deal with free iron. And it's also known that when it lives in this red blood cell, that iron chelators effectively kill the parasite, where they're really just cytostatic for bacteria and cancer cells. This is just a picture of an early ring form progressing to a hemozoin containing, or heme crystal containing, a scanning EM picture of P. falciparum heme crystals, and then an atomic view of the head-to-tail dimers of the crystals all by himself. So one of my first science papers looked at the role of this histidine-rich protein. You can see I call it the AHA protein. Some people laugh with it, ha ha ha. But very rich in histidines. Actually, histidine coordinates heme and hemoglobin. And right now, it's used for rapid diagnostic test. So I had mentioned earlier that one of the main pillars, three or four pillars of malaria control is an accurate diagnosis and effective therapy. So this HRP2 molecule does bind heme, but it is used in the majority of rapid diagnostic tests. I was also interested in not having malaria patients encounter a needle all the time to make a malaria diagnostics. Probably if you're sick, you don't mind, and it probably should be done right away, but if you're well, trying to eliminate malaria, you may want to look for a urine source. And if you look at urine or saliva, you're not encountering needles. And in Africa, there's not a taboo regarding using urine as opposed to provision of blood. So I approached a company making one of the malaria tests, and they said, sure, we can devise a urine protein test for you. And this was the first strip, the first patient being positive. Although soon after that, the company was bought out by a larger one, and they were not as interested in the urine malaria test, and it's taken a while longer to get going. But in the end, I don't think it's quite sensitive enough. There's about a third less HRP2 protein in the urine, and we need greater sensitivities. So we sort of thought about other ways to diagnose malaria. And actually, Ronald Ross saw malaria crystals outside the stomach of the mosquito and said, well, this is what's seen in the blood. The mosquito is then implicated. So some interesting properties about this heme crystal is that it polarizes light, it's paramagnetic, and it can absorb laser energy. So with colleagues at the Applied Physics Laboratory, we sort of demonstrated that you can just, without adding any matrix, you can just hit it with a laser and a mass spec machine, and hemazoin will give a characteristic signature 5-peak pattern where heme and hemoglobin does not, because it doesn't absorb the energy and ionize. And so we showed that we could detect it in a mouse infection. Miyang Nunt, one of her first papers, demonstrated that in microscopy-negative pregnant patients, that it detected more than half of them with this technique in the bloodstream, and that we could pick up all of the malaria species. So all we had to do was get blood, dilute it in water, and instead of having our eyes reading a glass slide, this was a laser reading on a metal slide malaria parasites. Although the machine to run it is tens of thousands of dollars and had to be reformatted. So that sort of sat on the table for a while. But then another researcher from Rice, this is his invention, although I did work with him on it. And I think it does have promise. I just want to point this out before I go to drugs. And so this is, when you remove a tattoo, you use like a nanosecond laser to break up the dye particles and your body absorbs it. So he got a very fast laser, a picosecond laser, that you can just put it on the skin. It comes up near a blood vessel. The malaria, hemazoin-containing parasite goes on by, and you make a picosecond laser, makes a bubble around hemazoin because it absorbs the energy, and the bubble, when it collapses, can be picked up by acoustics. And so here on the bottom, you sort of see before a malaria parasite in row A, and then after the pulse, you can see it optically or with an acoustic transducer. The malaria parasite is gone, but unaffected red blood cells are still present. He has demonstrated it in a single human malaria patient and in mosquitoes, but again, still some tinkering. One idea would be that we might be able to do it for airport screening, or it would be, well, I think it is a new dimension. There's still lots of things to be worked on out, but this hemazoin crystal can be used as a method for diagnosis. Back to drugs and how hemazoin crystals are made. So on histidine-rich protein 2, earlier we had shown with Dan Goldberg that it was present in vacuoles in the parasite where another histidine-rich protein was not. In a heme titration binding curve, that it bound heme, so it was present in the digestive vacuole, it bound heme, and importantly, we showed that it initiated crystal formation, and that that chloroquine inhibited the crystallization, and other molecules which bound heme, bovine serum albumin or human histidine-rich glycoprotein, did not generate crystals. So it's something unique to this HRP2. And we did Fourier transform infrared spectroscopy to sort of verify that we had the true heme crystals. But a criticism still was that P. vivax, mouse malarias, many primate malarias still make hemazoan crystals, and they don't have the HRP2 molecules. And one of the beautiful things about being here at the School of Public Health, years later, was listening in on one of Isabel Copen's presentations, and she showed electron micrographs similar to this, and sort of noticed with this malachite green staining the hemazone crystal coming out of a lipid area. So we're able to isolate some of the hemazone crystals. Did not use soap and water, but sort of had this film around the isolated hemazone crystals, where if I took pictures over on the side, they would, if I washed it with soap and water, SDS, delipidated it, they were the nice clear bricks, devoid of lipids. So went to basic cell biology and subcellular fractionation, where on a very dense sucrose or sugar cushion, very heavy, we were able to isolate pure hemazoin away from digestive vacuoles and proteins. And so we analyzed that and found, so here, showing that sucrose purified hemazoin did not have any proteins with it, trophozoites with lots of proteins. The lane is empty for a standard protein gel. And if you probe it with more sensitive Western blots, we see absent proteins there. But on a thin layer chromatography, we did see abundant monoacylglycerols in the last lane. So there were lots of lipids. And got some of those from Sigma. Well, we identify them with colleagues at Virginia Tech by mass spec, and sort of showed monosteric glycerol, monopalmitin were some of the abundant moieties in the sucrose purified hemazoin present in trophozoites and purified digestive vacuoles, but also there. So then we got them off the shelf from Sigma, looked at a blend of different individual components that were driven by our mass spec analysis, and showed that was able to crystallize heme, and that many of the individual ones also crystallized heme. So it sort of pushed it towards the lipid hypothesis. And if you think about it, it's not like a crystallographer here at the school making crystals. It's got to do it inside a water protein-rich milieu with an acid environment. So it has a lipid environment that the heme goes to that lipid interface under an acid environment and generates the crystals by itself. Part of the reason I was interested in how it made the crystals was because it was the target of the quinolines. So we knew that you had to be degrading hemoglobin to be affected by the quinolines, that liver stages or gametocytes weren't affected by many of the quinolines that we use. We also knew that the drug went to the lysosome like digestive vacuole. And this is an EM after low amounts of radioactive chloroquine that sort of shows three plumes right on the crystals, as if the drug is binding to it rather than being toxic all by itself. And so we showed that it was a saturable process binding to the crystals, but dependent upon heme. That drug by itself didn't bind to the crystals very well, but you sort of needed heme and drug to go ahead and bind. And in the late 2000s, Curtis Chong had spent about 6 months in Mozambique, and he was an MD PhD student, really wanted to work on malaria by itself. And like many of my summer students, I started him on a heme crystallization assay. And he used it a lot, and so much that my spectrophotometer, the light bulb broke. And this was like 2 weeks before his presentation. So then he adapted the assay to a plate reader, high throughput, and was able to run lots of conditions, and found out Well, one, he generated 2 notebooks about this much for his rotation, got a paper out of it, and but showed some interesting reversible kinetics of heme crystal inhibition, in that it took that it was time dependent and it was a reversible process rather than being irreversible, and that there's also a pH dependence to the heme crystallization. So before, this would have taken me weeks to generate the data, but with the new plate reader assay, we're able to get lots of this data fairly quickly. So in summary, right now we sort of have this process of making the heme crystal requires a pH less than 6. It can occur in a test tube with water, but in the parasite, we think it goes on in a lipid environment to exclude water so you can make the crystals. And you sort of make a head-to-tail dimer, then you form this larger crystal. And that inhibition is reversible with the quinolines, requires binding to heme, is pH dependent, and could either be that you're not making the dimer to go in or that you're binding at extra sites. So working with a crystallographer in Texas, sort of came up with this proposal on a kink site on a growing crystal. And so we sort of think that a heme drug complex binds into this growing site in red and sort of caps off the extension of the hemozoan crystals. And fundamentally, this is how the quinolines, which bind heme, work to kill the malaria parasite. And that's part of the iron problem. You just upset this a little bit, and you can make the best parasite in the world, which is the dead parasite. Curtis also wanted to saw a phenomenon of other drugs, ketoconazole, antifungals, working on inhibiting heme crystallization and the malaria parasite. So he said, well, why not go after all FDA drugs? And this is just a graph of showing what was available for purchase back in mid-2000 and known clinical drugs, a universe of around 10,000 drugs. So pretty much single-handedly, with a lot of medical students and help from my lab, he put together 2,500 drugs, which Laurent Shi and he and I screened for. This is one of my favorite bar graphs in the world of inhibition of the malaria parasite with 2,500 FDA-approved drugs. We found an antihistamine, a stemazole, which was very potent at inhibiting the malaria parasite, and showed that it worked by the same mechanism as chloroquine, inhibiting the growth of the crystals on this growing face. Although it was taken off the market, not because there were excessive deaths, but it's the most potent drug ever used in humans at inhibiting, at prolonging a QT interval in the heart, the human ether-a-go-go channels. And so it's kind of untouchable, although MMV did bring it up with me, but still something that I want to revisit because the drug itself was used in tens of millions of people for a couple of decades with no side effects. So one of the good things about being here in the School of Public Health is the history of our department. And so I was reading Lloyd Rosenboom's history and came across where one of the early chairmen, Hegner, had been quoted as that he had found a drug, hydroxyethylapocuprine, that was less toxic and could be quinine replacement, and that it was obscured by chloroquine and others after the war. But it had never been tested on P. falciparum. So we, with David Myers across the street, we synthesized hydroxyethyl apoquinine. It's sort of based on a quinine derivative. Showed that it inhibited lethal P. falciparum and also heme crystallization. And importantly, it had very poor this cardiac conduction channel inhibition. And that actually a quinidine derivative was more potent and in combination with artemisinase cured mice by itself. So still going back to some of the quinolines on finding rather than, so this was a drug, well actually hydroxyethylapocuprine had been used to treat pneumonia in Pittsburgh. It was in about 400 patients, halved mortality, but was sort of left behind because the sulfa drugs came along. In a way, chloroquine also sort of left it behind. But I think with the possibility of this delayed clearance phenotype, we think it's still possibly valid to move on forward and to explore some of the substitutions around the quinoline nucleus. So we had the FDA drug library, and we wanted, with Abhay Tripathi and Natalie Robinette, Abhay Tripathi had devised a cyber-green assay to try to look at drugs inhibiting metacytes on a plate assay. And his little trick was that we would add a quencher. So if the parasite was dead, it would take up a quencher, and only the live parasites could grow. So we had a good signal to noise, and so we were able to generate my second most favorite bar graph with gametocyte inhibition by the FDA drug library. And then there was an MMV collection that showed much higher hit rate, but they already worked against the malaria parasites. And we identified some known antidepressant like methylene blue maproteline, some antiseptics, chloroquine-like molecule, and we tested in a membrane feeding assay that they were potency at inhibiting oocytes. So they really killed the gametocytes, and so they're, part of more leads to follow up on. Well, so I've done a little bit of drug screening, and I sort of wanted to put that into perspective on where we are for screening for novel malaria drugs. So on the top, you sort of see where in 1880, Laveran diagnosed malaria in an Algerian soldier. Bird malaria is followed soon after that, and Ronald Ross demonstrated transmission of Plasmodium relictium of bird malaria. And then we sort of had malaria in mice and rats, Traeger and Jensen, 1976, for continuous culture. Malaria therapy for syphilis was in widespread use in the 20s and 30s. A lot of drugs were tried in humans then because of the experience clinicians had with malaria therapy. Ehrlich did the first drug screen with about 600 compounds, found methylene blue and salvarsan for syphilis by itself. And we screened about 14,000 compounds in the survey of antimalarial drugs during World War II. And later, Walter Reed added the collection to 2,500. Now there's about 700,000 Walter Reed drugs. And Roche looked at every new compound in the library against a hemazoan screen back around in the late 1990s, 100,000 drugs. I sort of came up with a more directed approach, and about the same time, about two other groups, Chris Austin at the NIH and Joe DeRisi, looked at these FDA drugs for blood stages. And since then, there's been about 3.5 million drugs that have been screened by two other drug companies, GSK and Novartis. So that's about 5 million drugs we've screened for malaria so far, but there's more than 10 to the 60th molecules out there. So we're really just a drop in the bucket, and we have lots of other molecules to look at by itself. But one of the problems with malaria, heme is sort of like iron. It can't be changed, and there's also been some observations regarding iron chelators and influence of iron on outcome of malaria by itself. So back to Christopher Murray, Back when he was a young child, his family showed that if you gave oral iron to Sudanese refugees, they had a higher percentage of symptomatic malaria. And then there was the famous PEMBA study, where iron and folic acid given to almost 25,000 kids in Pemba off the coast of Tanzania had a protective effect on children that were iron deficient by a zinc protoporphyrin IX test. No effect on children iron deficient with no anemia, but it increased adverse outcomes, hospital admissions, and deaths in children that were iron replete by the zinc protoporphyrin IX test with or without anemia by itself. So it sort of halted giving iron in malarious areas by itself. So how biologically was that possible? We sort of know that the malaria parasite living in the red blood cell is surrounded by iron. So how is a little 10 milligram dose going to really affect the outcome? We also know from test tube studies that extracellular iron has no effect, or iron status has no effect on growth parameters in the red blood cells. We know that there's like a 10 micromolar small labile pool inside the red blood cell, and there is some leftover iron and red blood cell ferritin, an iron storage protein. And, well, there's abundant heme and iron, but it doesn't degrade heme to release iron, but puts it all into the crystal by itself. So I had also worked on an iron transporter, which we localized to the plasma membrane of the parasite itself, saying that it was accessing this extraneous pool of iron that was pretty critical. This is just showing localization to the periphery of the plasma membrane of the parasite, and by a Western blot analysis on this iron transporter homolog protein by itself. And then there was, fate sort of does have its way. There was a student, Patricia Ferrer, her last name means iron in Spanish, came to my laboratory. And I hate to show a whole student's work in one slide, but in the summary, she had sort of worked with an iron chelator, first worked on blood stage malaria in the middle, and showed that you could remove essential iron, bioavailable iron, from a red blood cell, and then the parasite would be inhibited, and we could actually cure mice. When the iron chelator was gone, you could chelate bioavailable pools and sort of remove this essential iron source, and the parasite would be inhibited by itself. She recently has published with Yoel, also from our department, that the iron chelators also work on the gametocyte stages to prevent gametocyte to oocyst to the mosquito stage. And then we doubled back to the liver stage, and she sort of probed this interplay between hepcidin is sort of a master iron regulator. It's a hormone the body puts out as an acute phase reactant. And the end result is to restrict iron from, or bioavailable iron from the bone marrow and from some compartments, but other compartments in the body get excess iron with that. And so we sort of, on to the liver stage. It had been known for the from the Murray study, that if you give short-term iron right away, you have an increase in malaria by itself, or you have a low hepcidin. But you drive down the liver stage numbers in the presence of elevated hepcidin, or after a couple days of iron supplementation, your hepcidin goes up, and you also decrease this liver stage malaria by itself. So what we think was going on in iron-replete individuals on the island of Pemba, they would get the iron, have a transient protective state, but after a couple weeks, hepcidin would sort of normalize down, and then they would be more susceptible to the liver stage, to liver infection. And they would also be making more reticulocytes, and P. falciparum would have a worse outcome. So those are plausible explanations from this work to explain the adverse outcomes in the iron supplementation study. The bottom line is, I think we need to supplement iron in a malarious area with long-acting antimalarial, or just put them both in at the same time. And we know that treating both with iron and antimalarials, but the timing is very important because of this hepcidin molecule, that a lot of the iron that we give is not being absorbed because of this countermeasure effect on it. So I'm sort of going to close up returning to another antimalarial drug, artemisinin, which is a peroxide bridge, 2 oxygens, activated by iron or heme by itself. It generates free radicals, not necessarily oxygen radicals, but damages innocent bystander proteins. There's been a lot of popular press, a little bit of hype recently on its resistance, but the history of finding artemisinin goes back a little bit further. I mean, it was sort of the Chinese described Nueji on a tortoiseshell, described malaria. Yi Hong used this plant extract to cure malaria in 300 AD. During the Vietnam War, the North Vietnamese availed themselves to China for help with malaria. We're going to lose this war with the Americans if we don't have a cure for malaria by itself. And then a couple years later, Chairman Mao authorized Project 523, described in this book, where they screened 40,000 drugs and came up with the pure extraction from this plant. It was first tested in humans in rapid time. Actually, they sort of skipped over many animal studies. Actually, a plant extract was used in some of the human clinical trials. And this year, this Dr. Tu Youyou was given a Nobel Prize for the work during this critical period, and it really has been life-saving and turned around, part of the reason for the turnaround in malaria deaths. But we're sort of concerned in the malaria community. On that early graph, it sort of showed an uptick in deaths from chloroquine resistance, and we're wondering in Cambodia that there's been a description of delayed clearance to the artemisinin drugs, and does this really equate with resistance? I say it doesn't. It's not resistance, that it's treatment failure, but that's sort of another hot debate amongst malariologists, and I'm going to spend just a little bit of time going through that. So this Celts protein in a cancer model binds a transcription factor, and when it's altered or mutated, releases that transcription factor and can become oncogenic and affect kinases and drug targets. So work in the so we have a phenotype of a slow clearing parasites present on day 2 after therapy, and we've pinpointed it to alterations in this P. falciparum Celts gene. And the graph sort of shows that if you have wild-type parasites, you have a low prevalence of day 3 positivity, but if you have mutations in this gene, then you have a high prevalence of parasites present on day 2. Other people have characterized these genetic mutants that they have long ring stages and short trophozoites. Trophozoites are the stages that make the heme crystal. So it's just shorter to 12 hours rather than being about 24 hours or 20 hours. And the ring stages are much longer by itself. But there's many other reasons why malaria drugs don't work, or drugs don't work in period, besides the bottom, resistance to the pathogen. There's the wrong diagnosis. There's counterfeit drugs or poor quality drugs, not absorbing it, not being compliant, but also a suboptimal regimen, the dose, schedule, and duration of the drug by itself. So in contrast to the quinolines, which stick around for weeks with effective parasite kill, the artemisinin only last for about 6 hours, and A is a short plasma exposure with the artemisinins. And we also know to treat malaria, we're going after a short 3-day duration. So if you're treating tuberculosis, you're treating for 6 to 9 months. The half-life is 24 hours, so we sort of have this duration of drug exposure to doubling time of 168, triple figures. P-falciparum with the artemisinins, though, we are in single digits, barely 1. The quinine component, this ratio of duration of effective drug concentration to doubling time is in double digits in P-falciparum. In contrast to the short turnaround time of E. coli, we're in the hundreds for this duration of drug exposure over, so it takes a long time, a long ratio to effectively eradicate pathogenic organisms from the human body when they're causing damage by itself. So we had gone down to 3 days of the artemisinins, mainly because it was 90% effective. 5 days didn't add that much. 2 days of drugs was only 60% effective. So we sort of settled in on 3 days of drugs by itself, although the 3 days is a bit of a misnomer because we sort of count day 1, and then day 2 is 24 hours later, and then the third day up at the top is within this, in the blue line, 48 hours. So all 3 doses of our artemisinin drugs are under a single 48-hour life cycle. So it's like trying to treat tuberculosis in 1 day. It's like trying to treat E. coli in 20 minutes. It just doesn't work with a microbe, but we've been getting away with it with malaria therapy and the artemisinins. So a consequence of this single life cycle hit is the parasite said, well, I'll mutate, and I'll spend longer time as a ring, and I won't have the hemazone around or have the iron, and I can make it past these physicians trying to kill me with drugs if I just spend a short time as a trophozoite. So if you sort of take this first graph, superimpose it with the pharmacokinetics of the artemisinin drugs, you can see that if you're a drug-resistant or a delayed clearance parasite ring, and if we dose the drugs, we sort of miss the sensitive trophozoite in the solid line at the top. But the more sensitive, shorter ring stage does get a dose of drug on the sensitive trophozoite stage, releasing iron and making the heme crystals by itself. And so if you have this long ring stage, and if you sort of graph the kill rate over time, then the sensitive parasites are absent at 48 hours, but we still see these other parasites that are present. So we're still killing them, but I would still argue that we're dealing with a treatment failure and not drug resistance. And so that this 48-hour regimen is selecting for additional foci of drug resistance outside of Southeast Asia, but that if we just double the duration and do nothing else, that we can, quote, eradicate these delayed clearance parasites by itself, and that the trophozoites are still sensitive to the drug. And so we've sort of, it's also, well, we should be careful with terms. We have a ring stage artemisinin-resistant parasite. The whole parasite is still sensitive, but we have a ring stage artemisinin-sufficient parasite. We just can't go an extra day of therapy, but I think that we should double it on out. So Leah Walker in the lab is sort of proving this in an innovative mouse model, that if we do a multiple life cycle hit, it makes a difference in survival. And the blue curve is a prolonged survival with the artemisinin drugs versus the red dosing all the artemisinins within a single life cycle. And that in blue, we have a longer interval for absent parasites in the bloodstream by itself. And if we combine it with the artemisinin drugs, I don't know what happened with the MAC to conversion at the bottom here, but this graph is supposed to show single-dose piperaquine in gray doesn't clear the parasites, but if we do the longer interval multi-cycle same total dose of artesunate, that we get complete cure. But if we put all the artesunates within a single life cycle, combine it with chloroquine, that we only cure a third of the mice. So all drugs are dose and time dependent, and I sort of want to end, maybe the Brits had it right 200 years ago when they began to mix the malaria drugs with sugar and gin. It was much more concentrated back then, the quinine, but, and I guess it was easier to go for a longer duration. And present today, though, this tonic water, if I had a blue light, would glow. There's only about 20 mgs per liter of this. It was much more concentrated back then. We'd actually need 10 of these for each dose, so 30 of these in a day to drink all of this to cure malaria right now. And then while we're sort of on the subject of cocktails about to go, just want to highlight that James Shannon, one of the directors, founders of the NIH, is known for his research on malaria drugs. He picked up on fluorescent quinine and knew about it back then and developed many pharmacological assays for drug measurement in addition to testing chloroquine back during World War II. So I'll end there. I'd like to also acknowledge Lois, my wife, the best clinician I know, and my sons, my parents, David and Carol, my 4 brothers, many of the summer undergraduate students, PhD students, postdoctoral students, Master of Science students who contributed to most of this work, a very collegial, interactive faculty, and both in our department, Ron Gray's up there, and others who have helped me all along the way. And just like to say thanks. I'm glad, thanks for the recognition.
Thanks, David, for a great talk. So this is being recorded, so for your question to be heard on the podcast, I'll ask you to use the microphones. And that was a great talk, great slides, a great story. Let me just go back to the beginning where you're talking about the transdermal laser as a diagnostic test. I understand that creates a bubble, destroys the hemozoin in the cell. Is that right? In the red cell?
The hemozoin absorbs all the energy, and then it ionizes water around it and makes a bubble that then collapses back on itself. And you can pick that up. I mean, we sort of
right,
noted
but when
it.
you showed the light microscopy, the hemozoin was gone, right?
It had just been dispersed.
Dispersed. Okay. So it doesn't
so the
kill.
hemozoin, I don't think it actually. It was in a cell, and then that cell exploded, and hemozoin floated off. But the hemozoin was probably still there.
So I see. It destroys the red cell.
It makes a bubble, a small explosion, collapses back on itself. So you diagnose and kill at the same time, which is kind of nice.
Yeah. Yeah, that's what I was wondering. Could you use it, so if you had somebody with overwhelming blood stage malaria and you did extracorporeal circulation, could you kill, it's like it's not the public health approach.
You and Dimitri think alike.
Yeah.
He had the same idea, although I still would advocate a drug rather than filtration. We do exchange blood transfusion.
Sure.
Take out all the red blood cells and put them back in. But there's also hidden reservoirs. There's the spleen, there's sequestered parasites that aren't circulating, so you're going to miss those.
Yeah, it wouldn't kill everything. It just would kill the blood phase. Yeah.
Nothing like a drug to go everywhere in the human body rather than dialysis.
So I just want to get back to Chris Murray. So That was his father who was the first author? What was the story with the four Murrays?
Well, they actually wrote two papers. There were two papers, Murray, and that was the second. He wasn't on the first paper. I haven't met Chris, so I don't know, but he is an epidemiologist.
Oh, yeah, he runs the Global Burden of Disease
Right.
Study,
Right.
Institute for Health Metrics. Okay.
Right.
Any other questions or comments? No? No? Good.
All right.
Okay. So
we
we
should
have a
head
reception.
downstairs.
Where's the reception? Okay, Wall Wonder reception.
All
We
right.
can talk to David one-on-one.
Thanks.
Thank you for your work and for a great talk.
All right, thanks.